Yes, so to engineer T cells to suppress GvHD, we wanted to create a chimeric receptor that redirect these therapeutic T cells to specifically eliminate GvHD-inducing alloreactive T cells in their activated pathogenic state. Because in this way, we can prevent GvHD and also spare the majority of resting and non-alloreactive immune cells, and therefore reduce the intensity of immune suppression in patients. We then identified OX40 as a suitable marker to distinguish our intended target population, because OX40 is known to be upregulated on activated T cells, especially the CD4 positive subset. And this upregulation has been seen in many GvHD animal models and patients. In addition, previous studies have shown that using antibodies to block OX40 signaling pathway can effectively mitigate GvHD, and it doesn't impair the anti-tumor and antiviral T cell immunity. So we then engineered our T cells to express this OX40 specific chimeric receptor that enables them to recognize and then specifically eliminate OX40 expressing activated alloreactive T cells. We showed that these engineered T cells selectively target activated T cells, especially the CD4 positive population, but not the resting T cells or CD8 positive cytotoxic virus-specific T cells, both of which had minimal OX40 expression. Now, when we evaluated this strategy in vivo, we found that our engineered T cells could successfully protect mice against fatal xenogenic GvHD. And finally, we also combined this OX40 targeting chimeric receptor with a leukemia targeting CD19 CAR and showed that we can develop a single T cell product that simultaneously protect against both GvHD and leukemia relapse post-transplant. Similar to the on-target of tumor toxicity as we see in CAR-T cells, the one potential challenge of our strategy is what I call on-target of alloreactive T cell effect, because besides these activated alloreactive T cells, a subset of beneficial CD4 positive helper T cells and regulatory T cells may also upregulate OX40 upon their activation too. And then another potential concern is that the activity of our engineered T cells may exacerbate inflammation in GvHD patients. To reduce this risk, our lab is also developing engineered regulatory T cells that express the same OX40 specific chimeric receptor, so that we may be able to suppress alloreactivity without causing any additional inflammatory responses. And also because of these challenges I mentioned, I think it's very important to further evaluate our strategy in an immune-competent animal model that more closely resembles human GvHD pathophysiology before we launch any clinical studies. We have been collaborating with Dr. Viktor Tkachev from Dr. Leslie King's group at Harvard University to further evaluate our engineered T cell platform using their non-human primate GvHD model. And I think these studies will be very helpful in the future to guide our decision-making process during clinical translation.